two input frequencies, one low, one high, and the higher frequency must be 11 times the lower. It's what we musicians would call the 11th harmonic. And when we add the 11th harmonic, we begin to shatter microorganisms like a crystal glass. These are the first videos taken. We showed these videos to our friends in the biology department. They said they hadn't seen anything quite like it. Seems to be a new phenomenon. These organisms are being shattered by our electronic signals. This is a harmless organism, almost friendly, a little blepharisma. And normally they're very fast swimmers, but when you approach a frequency to which they are vulnerable, they begin to slow down, then they stop, and then they begin to disintegrate within about 3 minutes. So now we know we can destroy a microorganism, and the question comes up, but can you target a specific organism with a specific frequency? So in this next video, you'll see a large organism in the center, a paramecium, undergoing disintegration, and swimming all around it, a tiny different organism, which is unharmed. If we're lucky, we'll also hear the audio as I narrate the experiment live in a noisy lab. Perhaps you can see this sort of fireworks effect happening in the growing blister to the right of the organism. And here comes a little neighbor, wondering what's going on. Who's testing. And you can see blisters forming now on the lower left quadrant and upper left quadrant. The shape is now changing. And a major explosion out the top. So now we have some evidence that we can target specific microorganisms with specific frequencies. And we make several more videos, and we film the destruction of hundreds of microorganisms. And about this time, we meet a cancer researcher, and we show them these videos. And this results in an invitation to spend 4 months in a cancer research lab trying to shatter cancer cells. This is our setup in the lab. You can see the microscope with cancer cells on it. Here's the plasma tube, and here's my little frequency control box. So first we attack pancreatic cancer. Take a good look at this slide, because the next one will look quite different. After we treat these cells, they change their shape and size, and they begin to grow long, rope-like structures out the sides. They look something like antennas. I call them bioantennas, for biological antennas. It's as if the cancer cells are trying to tune in to our signal. It also turns out this is the beginning of a process of destruction for cancer cells. We now know that cancer is vulnerable between the frequencies of 100,000 hertz and 300,000 hertz. So now we attack leukemia cells. Leukemia cell number 1 tries to grow a copy of itself, but the new cell is shattered into dozens of fragments and scattered across the slide. Leukemia cell number 2 then hyperinflates and also dies. Leukemia cell number 3 then tries to make another cancer cell. The new cell is shattered and the original cell dies. But killing a handful of leukemia cells is not enough for a patient. What kind of numbers can we do? In repeated controlled laboratory experiments, independently assayed by the 2 top experts, we killed an average of 25% to 42% of the leukemia cells, as high as 60%. They also determined that we slowed the growth